Every time you remember something, your brain has to save it again, and for a few hours, that memory can be edited. Welcome back to The Explainer. Today, we're completely challenging everything we thought we knew about how memories are actually stored.
I mean, it sounds like total science fiction, right? But this is a monumental shift in our understanding of the brain. For decades, we were taught that once a memory forms and consolidates, it's permanent, written in ink, basically. But what if simply bringing a memory back to mind actually turns that ink back into wet paint?
Okay, so let's dive right into this. Think of it like unlocking a secret vault in your mind. This temporary window where a recalled file in your brain is basically unlocked and vulnerable is known as memory reconsolidation. When you pull up a long-term memory, especially a fear memory, it doesn't just sit there perfectly intact. It actually returns to an unstable, or what neuroscientists call a labile state. And to keep that memory, your brain has to actively restabilize it. If that resaving process gets interrupted, well, the memory itself can be altered or maybe even erased.
Which brings us to Section 1, the biology of the resave, starting with the animal model.
Back in the year 2000, researchers Nader, Schaffe, and Ledoux made this absolutely wild discovery. They found that saving a memory actually requires protein synthesis. So, by blocking protein synthesis in the amygdala, specifically during that brief reactivation window, they actually weakened the fear memory. This was huge. It gave us direct evidence that restabilization is an active, disruptible biological process. But, okay, we need to plant a massive flag right here. This foundational work is an animal model. We're talking about rats, not humans, rodents injected with protein blockers.
So let's move to Section 2, a drug-free update, or what we can call behavioral hacking.
Scientists started wondering, could we rewrite this fear without literally injecting protein blockers into a brain? Could we use behavioral techniques alone to hack this biological resave process? The absolutely crucial point here is understanding the difference between the traditional approach and this new frontier. Standard extinction, which is the whole basis of traditional exposure therapy, basically just covers up the fear. It layers a completely new, competing safe memory right on top of the old fear memory. The problem? The old fear is still under there, just waiting to pop back up when the context changes or time passes. But reconsolidation updating, that theoretically alters the original fear memory trace itself during its unlocked labile window. It actually rewrites the underlying file so the fear fundamentally changes and doesn't return.
Fast forward to 2009, researcher Marie H. Monfils and her colleagues tested this idea with a pretty brilliant behavioral hack on animals. The timeline basically works like this. Step 1, trigger the fear with a single brief reminder, just enough to unlock the file. Step 2, wait for the reconsolidation window to open, letting the memory become unstable. And then Step 3, provide standard extinction exposure, repeated safe experiences, while that file is still actively unlocked.
And the results? Honestly, striking. Doing the extinction training specifically inside that 10-minute to 1-hour reconsolidation window prevented the fear from spontaneously recovering far, far better than standard extinction performed outside the window. By timing the safe experience precisely while the memory was actively resaving, they essentially overwrote the fear. But again, keep in mind, this was still an animal model.
That leads us to Section 3, Human Trials and Hope. Translating all of this to humans.
This incredible animal data really set the stage for bringing this retrieval-then-extinction procedure to human beings. Could we actually hack the human reconsolidation window just by, changing the timing of therapy? Well, a landmark 2010 study by Daniela Schiller and her colleagues tried exactly this in a lab setting. They used a brief reminder of a condition cue, waited for the reconsolidation window to open, and then followed it up with extinction training. And the results were stunning. This entirely drug-free behavioral procedure successfully prevented the return of fear. No renewal, no spontaneous recovery. It seemed to completely confirm that human fear memories could be fundamentally rewritten just by tweaking the timing of exposure.
But we have to move to Section 4, the critical honesty beat, a necessary reality check.
Look, it sounds like the absolute ultimate silver bullet for anxiety, trauma, and phobias, doesn't it? But as your explainer, we really need to hit pause right here. A major 2020 verification report by Chakia and colleagues failed to cleanly reproduce those 2010 human results. That's the messy reality of the scientific process. Independent replications of this retrieval extinction effect in humans have been decidedly mixed. Some labs find the effect, and others simply do not. The human brain, with all its crazy complexities and variables, is just much, much harder to precisely hack than a rat in a controlled cage.
So I want to be absolutely perfectly clear here.
While memory reconsolidation is a fascinating, genuinely promising mechanism, it is actively debated right now. It is not settled clinical practice, and it is certainly not a validated therapy for children. Almost all the causal, really mechanistic evidence we have comes from rodents, and the human evidence relies on small sample laboratory conditioning, not real-world clinical trials. We cannot simply declare the problem of fear solved.
Finally, Section 5, our Fear Me Not takeaways.
Now, despite the messy reality of clinical replication we just talked about, the underlying concept here offers a really powerful framework for our Fear Me Not approach with kids, and even for our own anxieties. The biology points to something profound about how learning actually works. It suggests that when we pair a child's scary memory with a fresh, brave experience, the exact timing and the specific way that memory is reactivated might be just as crucial as simply repeating the exposure over and over. If we can trigger the memory gently, open up that window of lability, and then introduce a powerful, safe, and brave experience, we might just be stacking the biological deck in our favor, helping the brain literally integrate that new safety into the old fear file.
If you want to dig a little deeper into the standard building blocks of layering brave memories over fearful ones, make sure you cross-reference this with our previous explainer, number 86, on fear extinction. Memory is not a tape recording, it's a reconstruction, and every single time we reconstruct it, we just might have the power to change its ending. So, what old fear file are you going to try to rewrite today?